thanks to the Centre for Personalised Medicine and the Wellcome Centre for Human Genetics, and to Tim for giving our plenary lecture at the end of a fantastic day of talks and lectures. Thanks, Tim. So Tim and I go back some ways. I was looking back at the 1,000 emails I've saved over the years, and it began in 2005, as many things do, by Tim trying to get funding for his twin study. And like all these pathfinding studies, ALSPAC, even UK Biobank, their origins stuttered in the quest to get funding because people weren't very keen on cohorts or long-term studies, which turned out to be vastly incorrect. We did a Mendelian randomization study together on vitamin D, but with one genetic instrument, and thankfully it never got published, Tim. But more recently, the great George Davies Smith stepped in with the latest genetics on vitamin D and type 1 diabetes, and did a more thorough analysis, which was published. Tim and I have, as I said, have crossed paths on many occasions, including very keen interest in measuring the metabolome, for example. And I always got the impression about Tim that he'd never fulfilled all his ambitions, and he wanted to be really, really famous. And all of a sudden, in the last couple of years, he has achieved exactly that. So the PREDICT study looks fantastic. I'm sure he'll explain what the Messerius Zoe is. Paul Franks gave us a very short preview earlier in the day. And the PREDICT study, and which Tim will tell us about, is exactly the sort of approach you need to take. It's ambitious, it's complex, but it gives us key information. And then, of course, his other claim for fame, which I guess he's got his OBE for, is his role in his app in the pandemic, and his subsequent 69,000 followers. And Tim, you really are making key contributions now, in addition to your long haul on the twins. So congratulations on that, and we're really looking forward to your talk. Thank you.
Thanks very much, and thanks everyone, and thanks for inviting me. It's a real pleasure to talk about my latest hobby, which for the last 10 years has definitely been nutrition and the gut microbiome. And I'm going to try and rattle through quite a lot of data, but I'm hoping that we'll have plenty of time for questions, because in a way the questions are often more interesting than the pure data. I'm just checking whether this is in the right mode. Okay, everyone happy with that? Good. Well, so my title is Professor of Genetic Epidemiology at King's College London, and for transparency purposes, I'm a scientific co-founder of the company Zoe, which is a sort of King's spin-off that you might have heard, probably more in association with the COVID symptom study app, which has 4.5 million downloads and about 1.5 million people using it still, which is amazing after a year, which I'm not really going to talk about other than to say it's an amazing health device that I think is going to transform the way we do research in the future, because the speed at which we've done it, and we could discuss in questions some of the areas that come out of that, like we did a study of 1 million people's health habits and diet and exercise in the space of 2 weeks using this app for virtually no money at all. So when you put those in scale, you see how this might change all of our research habits. But my career, as John said, actually started a long time ago, about over 25 years ago, when I started studying twins and have spent my whole life scraping the money together to keep this cohort going of now 14,000 twins, half identical, half non-identical. And it's the largest UK cohort, and it's certainly the most detailed studied group of individuals probably on the planet. Many of them have been with us from the start. And where my studies took me was really firstly on the genetics, showing how similar they were. Isn't everything genetic? Isn't that amazing? To exploring the genes, particularly in the non-identical twins, to look at diseases. But I think the last 10 years, I've been much more interested by why identical twins were different. What makes one be depressed, one happy? Why is one overweight, one skinny? Why do generally they die of different diseases? Why do they get very different cancers? And all these kind of questions kept coming up in my mind, which is kind of odd because these were genetic clones. And apart from a few rare mutations that all of us have, essentially identical in every cell in their body. And the big thing that took me on my path towards the gut microbiome and nutrition was the fact that twins ended up having very different microbiomes. And they was radically different, not just subtly different, like you might find with epigenetics, but radically different. And to me, this suddenly explained this link between nutrition and health in a way we hadn't really thought about before. And that's why I wanted to start getting into large-scale studies. And I've just got a picture of the COVID symptom study app. If anyone hasn't seen it and downloaded it, please do. It's particularly interesting now in the vaccine era. And we have other data we're going to release on nutritional studies in a way that might lead your state of nutrition and COVID and vice versa. Anyway, back to nutrition. And what's interested me, and each time I've gone away and to research something, as I've changed my areas from epidemiology, rheumatology, genetics, genetic epidemiology, epigenetics, and for the last decade, nutrition, I've gone away and written a book on it. So it's a good advice for anyone out there who wants to really understand a field is basically, it's like writing a thesis. You go away, you hide for 3 months, write a book, and you come back an expert. I don't regard myself as an expert, but because I don't think you ever get full expertise, as we see here, because in the field of nutrition, everyone is an expert, and everyone eats food at least 3 times a day and think they know much more about it than anything else. And we asked 13 professors of nutrition in UK and the US to rank 105 food items, common food items, in terms of a 0 to 10 scale on whether it's very healthy, very unhealthy. And this is what we'd see with perfect agreement. And you can see this is what we saw in reality. And that really was quite interesting because everyone agreed that eating biscuits and KFC is not very good for you. Everyone agreed that eating blueberries and olive oil were good for you. Virtually none of them agreed on whether raisins, dairy, yogurts, cheeses, orange juice, or even Diet Cokes were good for you or not. So clearly there isn't as much agreement as we've been led to expect as the standardized one-size-fits-all dietary guidelines suggest. Now, some of the reason for that might be just the way that we've addressed nutrition, which I think is, for those like me that went through medical school, it was seen as one of the simplest areas of medicine and one of the least intellectually demanding, and that's because it was all just about a few key nutrients, and most students still learn more about scurvy than they do about obesity because there's a nice simple mechanism there. But we now know that food is much more complicated than we thought. Rather than just 3 macronutrients, we know there are at least 34,000 metabolites all interacting with each other, combining with our own personal metabolites and our 20-odd thousand genes, and of course our 2,000 species of microbes and all their 200-fold genes. So it is complicated. So if you take that complexity and then you look at the simplicity by which most nutritional research has been done in the past, you start to see a pattern. And a good example of this is an excellent study, in my opinion, called the Diet Fit study, where the group in Stanford were paid a lot of money to try and, in a way, show that a low-carb, high-fat diet was going to be better than the opposite high-carb, low-fat diet. So they took around 609 overweight Americans, 60-year-olds, and put them on a healthy diet, so plenty of fruits and vegetables and good advice, but they changed the proportions of the fats and the carbs. And at the end, there was no difference between the groups. So a lot of disappointed people in this area because people had sort of banked their reputations on this one way or another. Everybody lost weight, about 5 or 6 kilos on average. But this is where it gets interesting because these are called waterfall plots, and each one is an individual. And you can see that whereas the mean was this minus 5 kilos, some people actually gained 10 kilos in both groups, and some people lost 25 kilos. So it could be that your choice of which one might suit you could end up with a very different result. No one had really looked at individuals before. So that's one reason why we did the PREDICT study, which was published last year here, but was essentially 4 years of work, which came about with a meeting between myself and 2 entrepreneurs to set up the company Zoe, and they promised to actually provide funds to do scientific research before launching anything. That was the deal that I set. You said plenty of companies start nutrition projects based on hot air and marketing. Let's get some real scientific studies done. And amazingly, they went away and they're really good at raising funds. And they, within a few months, had the money to start the first PREDICT study, which we call PREDICT 1, which was 1,000 people, mainly twins, but a few in the US. And we'll go into the details of that. And of course, this is just a whole series of these studies. We've only published the first one. We did virtually a copycat one in the US, which was home-based. And we have all these other sub-studies here, which in time will be published as well. And the PREDICT 3 study is actually 10,000 people. The first 10,000 people are actually paying customers. And so they're paying for their own study and they're agreeing to do research. So the numbers are growing rapidly. But very briefly, because this has been published, but I know some of you might not know the details, the idea was to do the most comprehensive study to date, looking at individual responses to food, because we've never really looked at what happens after a meal. Everything's been done in the fasting state, whereas we spend most of our lives in a postprandial state. So all 1,000 individuals came either to St. Thomas's or Mass General, and a whole series of bloods were taken on the day, metabolic challenges, metabolomics, DEXA scans, genetics, blood pressure, questionnaires, etc. And then they went home with a continuous glucose monitor to record their blood sugar for the next 2 weeks every 5 minutes. And they were logging all their foods. They were given wearables to look at exercise and sleep. And we had lots of blood tests to look at things like triglycerides and inflammation levels and metabolomics, and of course, stool samples. And the response was amazingly good. Even by the twin standards, people loved this study. They absolutely loved it. Now, the first really wow thing that we saw was that these are normal people. They weren't selected for disease. They were healthy enough to come and have all this stuff done on them. But what we saw was an 8-fold difference between the responses in the blood of individuals to an identical meal. And this is a standardized meal, and you can see the lipid levels changing, the glucose and the insulin. Nobody was the same. Everybody, in fact, was unique. And that was the first thing that really hit us, that there is this amazing variation that's never really been looked at. And of course, having twins allowed us very quickly to get a handle of what it would look like if we were measuring height. This is what we'd see. We saw with this glucose response a rather weak effect, around 30% heritability, and virtually nothing when it came to triglyceride response. So rather different to what we've been seeing at the baseline. And when we put all these together, we saw that this variability was definitely not explained by genetics. It wasn't explained by any one single factor, a combination of them. Meal composition was one of them. Context was important, baseline markers, lipids, microbiome, and a slightly different pattern when we looked at the blood lipids. And all of them showed a multi-factorial approach to this, which meant that our responses are much more complex than we thought. We also started playing with the data because we had so much of it, and we still haven't explored most of it, but up to then, most people have been looking at sugar peaks, glucose peaks, and generally within a 2-hour window. But some guys in our group who weren't actually medical at all thought this is a good idea. Let's look a little bit longer. Let's look at 3 hours and see what happens. And it turns out that they saw certain individuals after foods had a sugar dip after 3 hours, and that was correlated with a peak, but the dip was actually much more predictive of various items here. So people in the quartile with the biggest glucose drop had increase in hunger, reduced alertness, they ate their next meal faster, and they ate more calories, about 300 extra calories in that day. So an identical meal in some people will give a very different response and make them eat more. So again, as well as being interesting from a physiological point of view, it makes a mockery of the idea that all calories are equal. We also, this is just ongoing research showing that we can now pick up what happens at the 6-hour level after you've eaten a meal. Some people, this triggers an inflammatory reaction. In other people, it doesn't. So there does seem to be differences between these inflammatory markers that's going to start affecting what's going on. But our basic premise was that these sugar peaks or these lipid peaks postprandially are generally bad for you long term, although everyone has them to a small extent, but people who have excessive ones cause a stress to the system that changes your metabolism. And so everything we've been doing is to try and modify those to reduce them, to reduce inflammation, reduce stress in the body, reduce fat deposits. Now, the second part obviously was to go into more detail in the microbiome. The first paper just looked at 16S, didn't do any detailed metagenomics, but now working with fabulous Nicola Segata in Trento with the latest bioinformatics tools, we really dissected what was going on in the gut microbes and see how they were important for food. And first thing we confirmed, what we'd seen in the 16S data is that no two people are the same microbially, and as opposed to genetics, we only share 25% of our gut microbes. And even identical twins only shared slightly more than you'd expect. And we all have unique microbiome strains. And a sample of your gut microbes is, in my view, a better predictor of health than our DNA. And of course, it's probably the chemicals that the microbes are producing that's key, and they give that unique chemical signal. But going back to the paper, we were the first to link the microbes with foods in a systematic way, and also link that with health. And this is just an example from the paper, which came out in January, if people want to look at it. But heat maps showing that certain ones, like you'd expect, like yogurt, were picked up here, but also other favorites of mine, like dark chocolate, red wine, and then negative ones that we think ketchup and sauces and processed foods are not particularly good here. So we were able to pick these associations between the microbes and foods, and then put these into groups. And interestingly, it wasn't just plant-based versus animal-based. We were able to see these groups of microbes that were associated with unhealthy plant and animal-based foods as well, showing that the overall dietary pattern is probably crucial as well, not just individuals. And this allowed us to get this overall signature of the healthy microbiome, produced 15 good bugs, 15 bad bugs. And you can see that we measured all these different health parameters here, whether it was your ratios of your lipids, your blood pressure, your cardiovascular risk score, your visceral fat levels, or your inflammation levels. Everything seemed to correlate into these groups. So we're now starting to use these 15 good and 15 bad bugs as a really good snapshot of how your microbiome is doing, because these are there's a few sets of microbes that are common to most of us. And this is just another way of looking at it. Basically, these are the summary of what I've just shown you. Now, the idea, of course, it's fine to have these academic papers. It's nice to have papers in Nature Medicine, but if we can actually start to change people's behavior, then that's the key to this. And machine learning allowed us to get individual predictions about, after these test meals in that first 2 weeks, about how you would then respond to any food. And these models got around 80% predictive ability, and we then created these scores on the basis of that. And this is the Zoe program. This is a bit of a blurb from the website, but introduces this idea of understanding your own personalized scores, doing your own experiment, which is essentially a home version of what we did in the PREDICT studies. And we've now rolled this out to several thousand people in the US. It's been going for about 3 or 4 months in the US, and we're starting to get feedback on those results. But these are my results, just for transparency, and they're not very healthy. I've got dreadful sugar result here, and dreadful fat control. The only good side is that I've got a good balance of gut microbiome, thank God. Otherwise, no one would believe my books. And with this result, putting these things together into this algorithm with other components about myself, the company gives scores back based on all these criteria. So this is my personalized score. So I get only a score of 23% for bananas. And I used to think they were super healthy foods. So I'm much better off eating an apple than a banana, whereas for other people, it really doesn't matter that much. And I certainly changed many of my eating habits since getting those back, changing my breakfast, for example, in quite a big way, and reduced carbs. Another bit of good news, I needed it, was that I was one of the 31% of people in the UK and the US who have a nice parasite. It's a friendly parasite called blastocystis, which we thought was only associated with travelers' diarrhea, but it turns out that this is strongly associated with better metabolism of fat and reduced internal visceral fat levels. So this is only possible through the metagenome analysis, and we're discovering many new microbes and parasites every few months, all of which have these functions we couldn't have dreamt about. So it's very important to keep an open mind. And these are the personalized gut boosters. I was recommended to improve my ratio of good to bad microbes from that list of 30. And everyone would get slightly different ones. And this list will constantly change as we understand more and we get more data. So in summary, everyone is unique in their food response, even identical twins. And genetics only explains a fraction of how we respond, which was a surprise to me, given my lifelong experience in genetics and twins. And the other shock was the composition of the meal explains less than 30% of our overall responses. And that's really what we see on the label of foods. And so all this idea of understanding our food response based on what a supermarket label tells us about calories and the macronutrients of what we're eating turns out to be much less helpful than we've been led to believe. And the gut microbiome is a key player here that can be potentially manipulated by our diet to improve our health. And I think we have quite a lot of data to show. There is another slide I might be able to find from looking at the first few hundred participants in this program that are reporting back they're feeling very motivated by this data in a way that wasn't seen with genetic type information that was discussed earlier. So we're seeing big improvements in people's response to, they're saying they have more energy, they're losing on average about 8 pounds in weight, and they are feeling less hungry just by doing some of these nutritional swaps. And I think a lot of this is because of the instantaneous nature of the personalization. The glucose monitor is put on, and for any of you who haven't seen it, you do get a real-time readout that allows you to see exactly what's going on when you're drinking orange juice or you're having your sugary muffin. And this really is not something that is just going to happen to you in 10 years' time. You can see this now. So this is why when so far about 80% of people have then, after taking the test, have gone on to do a 4-week plan where they are monitored as they go through this swapping of the foods based on their food scores without changing calories, without skipping meals or anything. And so far the results are extremely good. We obviously are setting up trials, but at the moment, just looking at what the observational studies are showing us and what the feedback is. And clearly, the other point is that many people are requesting retesting in 6 months' time, which suggests they are happy with the results and want to continue to engage with this kind of personalization. And I think the fact that we have now moved this from the research arena to a commercial arena means that we can scale this up 100,000-fold, and the company is currently doubling its what it can do every 3 months. And so within a couple of years, we should have several 100,000 people's data and be able to get much more granularity on many of these associations. It's currently available in the US, and hopefully will be here in the autumn. And the great thing is everyone signs up to share their data with researchers, and we're committed to publishing all this stuff. So I want to just acknowledge the team of several hundred people that are behind these projects, of which I just get to be the spokesman. Particularly want to highlight at King's College my colleagues in the Twin unit, but Sarah Berry, who's the key nutritionist that has been a fantastic colleague to work with. The whole Zoe team, of course, and there's now nearly 100 of them in Boston and the UK, and colleagues at Mass General, Harvard, but particularly for the microbiome work, Nicola Segata and postdoc Francesco Asnicar have been amazing and give us the most up-to-date data. So I'll stop there and hopefully take some questions.
Tim, that was fantastic. Congratulations. So we got a few questions here. One is, I know you've been pretty outspoken about healthy diet versus pro or prebiotics. Could you, that's the first question. What's your view on that? Lots of people spend lots of money on probiotics and prebiotics.
Well, if you're healthy and you have a varied diet, you should be getting all the probiotics and prebiotics you need. So that's what we should be encouraging, rather than selling people expensive formulae, because it's a bit akin to the vitamin supplement market. The more people talk about vitamin supplements, the more families start buying it regularly, the less they worry about giving their families nutritional food that's diverse and covers all the vitamins. So you get into a bit of a vicious circle. So I'm against it as a concept, as well as scientifically. So it's not to say that I don't think they work. And I think the evidence are that in general, and we've done some meta-analyses on it, in general, probiotics do work for many conditions, although there's absolutely no agreement about which probiotics work, because everyone uses a completely different one that's patented and it can't be used in another study. So there's a real problem there. But the other problem with probiotics is you're only getting a few microbial species or strains, and as we're all unique, we're going to respond very differently to them. And that, again, you might start to need personalized probiotics. And that's probably why we're not seeing as good a results with those as we should do. And until we end up with multi-probiotic supplements that have hundreds of them, I think we're going to be slightly stuck there. But I think there are certain conditions, people on antibiotics, people with gut disorders, people with inflammatory disorders that can benefit from them. So absolutely, I'm absolutely not ruling them out. I just think we should first make sure that our population has a healthy diet that is not full of processed foods, that has a diverse range of gut-friendly foods. And if you've got your microbiome as healthy as you can, and you still need something, that's when you might need these boosters. But other data is clearly showing that prebiotic supplements like inulin, etc., don't work as well as say a Mediterranean diet, because they don't give that same diversity. And if you just give a prebiotic, it will grow a certain type of microbes only, but won't give you that diversity that gives you this metabolic diversity. So that's my general principle about this. Let's get everyone's microbiomes healthier first with 30 plants a week, fermented foods, cutting out ultra processed rubbish, and generally eating better.
Yeah, so we're just starting a massive randomized control trial to prevent the autoimmunity in type 1 diabetes, which begins in the first months of life. It's been funded by the Hemsley Charitable Trust. And it is a minefield of which probiotic, but it sounds as if your recommendation would be to give mums who are pregnant, make sure they're on a healthy diet, rather than giving them a pro or prebiotic or a combination symbiotic, and get mums' microbiome and diet as healthy as possible.
That would generally be, yes, that is my view. And I mean, there are some exceptions with infants, and there's bifidobacteria, infantalis, that are specific to neonates, that usually the natural birth and then breastfeeding, etc. If that's lacking, then I think there's a good case for those type of probiotics. But in general, let's try and improve everyone's health through these natural fertilizers and these natural probiotics, and get the whole of the UK having more fermented foods without sweeteners and additives and the other rubbish. And many other countries have done this naturally for centuries, and we've just the Scandinavians do it, Eastern Europeans do it, Indians do it. Everyone has got their fermented foods as part of their culture except us, and we just need to get it back and throw away all those unhealthy children's yogurts that are just full of sugar, and get back to the real thing.
Yeah, I agree. And another question is, are there some foods that are universally beneficial to almost everyone? So the person has suggested brassicas, or is there a killer app food?
Is there a killer vegetable? No, I mean, most vegetables are healthy for everyone. No one's really found anything wrong with them. Obviously, there's always might be somebody with an allergy that's rare to it, but in general, the vast majorities, particularly of vegetables, are healthy for everyone. Fruits, there are a few that you can get allergies to, but in general, if you discount the allergy, I think in a way, a diversity of plants in moderation is generally healthy for everybody. The idea that there is some group of superfoods and other ones that are not, I think is really debated. But clearly, I like to quantify the foods by not so much sugars and fiber anymore, but looking at polyphenols. And I think this is very much the future of foods, is by ranking foods based on their polyphenol counts. These are the defensive chemicals in plants that we now know actually feed your gut microbes. So they're the fertilizers for your gut microbes. So that tends to be the bright colored plants, so that a red cabbage has thousands of times more polyphenols than an iceberg lettuce, for example. So there are some very boring vegetables that don't help anybody. They just fill up your plate and make you think you're eating healthily. But if people focused on the colorful, high polyphenol ones, then I think that's a natural remedy everyone can have. And my view is we should be having 30 different plants a week, based on a study we did with the American Gut Project, in 11,000 people from the US and the UK, that showed that seemed to be the optimum point at which you had a maximum gut diversity. And that includes nuts and seeds and herbs. So it's not as difficult as some people think.
Right. So those 30 things are a great variety from red cabbage to seeds. And since we're hosting this in Oxford, we think we're good at randomized control trials, Aidan Doherty is asking, are there plans to use the platform in a large-scale RCT? And if so, what would you select as the primary endpoint?
So I think what we probably, it depends really what people want to use if we're talking about the commercial product of this. Most people at the moment seem to be using it for weight loss. But there is a group of people that are just using it because they want to feel better. Their weight is fairly optimal. They just want to feel they want to have more energy, and they might be worried about future cardiovascular problems. But I think if we'd probably have weight loss as the primary endpoint, and appetite and energy levels as the secondary ones. But we're keeping our minds open on that until we finish, in a way, the observational study to see what those first results are. Because we were surprised about these responses in energy and tiredness. People who are switching, perhaps from carbs to fats or vice versa, just reducing the number of sugar peaks they're having in a day, we're reporting, without knowing what they were, we're definitely reporting greater energy levels. So I think that needs to be taken more seriously than we've done at the moment. And there's, as well as appetite, there is very little work on that on large scale populations.
Yeah, so people are obviously very impressed with what you're going to do with Zoe. And I think you answered the question that Zoe will come to the UK at the end of this year. Is that correct?
Before the end of the year, yeah.
Before.
We're hoping sometime in the autumn. We've got lots of demand, and it's just logistics, and COVID has delayed us. We have to get all the labs set up, and it's quite an interactive process as well. So we're going to send the kits out, people doing their own experiments, and they get the results back, and then there's an interaction with teams of nutritionists, etc. So it's quite a complex process, but we're getting there. So bear with us. For those of you can go to if you're not already, go to the website, and you can join a wait list. That's joinzoe.com.
Wow. Okay. And let's say I haven't got that beneficial parasite, blastocystis. What's your view on me, or somehow being able, or somebody else that hasn't got the parasite, to get it, to specifically implant the parasite?
I think that's an interesting question. And of course, we are going to have a group of these, and the other sort of big microbe group is Prevotella, which keeps coming up as being useful. And the question is, if you don't naturally have it, is it going to stick in you, and is it going to have the same benefits as it would in someone that's not naturally there? So I think we need to do some trials of this, and I think it's going to be more complicated than we think, because to have blastocystis growing in you, you might need a specific environment, or you might need to be surrounded by other microbes that produce the right chemicals for it to really thrive. And I think so it might be a combination of giving it the right food, but also having a community around it that's providing the other metabolic byproducts. But I think we need some brave people like you, John, that's going to volunteer, a bit like Helicobacter, to swallow some blastocystis and see if you feel healthier. So maybe that's what Oxford could start doing.
Yeah, I know some of the fetal transplant studies that are published are really dramatic, but I'm not a big supporter myself. There are people around, including in my laboratory, that would say it's all down to bacteriophage, and they control everything. Presumably, you're reading out on phage prokaryotic viruses as your metagenomic sequencing gets better and deeper and more affordable.
Yes, I mean, there are roughly 5 viruses or 5 phages to every bacteria.
Yeah.
And so we've got 100 trillion or so bacteria. So that's 500 trillion viruses. And it's generally believed they keep the bacteria in check and actually act as a balance sort of predator to stop, one group getting too big. So we know very little about them, but I think in 10 years' time, we'll know a hell of a lot more about them and whether we can start to manipulate viruses in the same way. But I suspect that we've got our hands full just working out what all these bacteria which we're sequencing are and all these novel functions. And my guess is that they're going to be more important generally in the normal state. I think in the disease state, these phages might become really important and might have roles in cancer and other things like that. But I think before the viruses, we probably also need to, as well as these parasites, the fungi are the other area.
Yeah.
Play a really big role in inflammation, and there is some evidence that they would play a role in colitis, etc. And I think we just need some better methods to crack open their DNA and start really looking at them. And I think that'll be fascinating. And then we'll start getting nutritional advice to build up the good fungi and reduce the other ones. That's what I think is happening. But yeah, there's plenty to do, that's for sure.
So that brings me on to eukaryotic viruses in your gut and the interaction with the other residents of your gut microbiome. Presumably you're collecting eukaryotic virus information as you do this metagenomic sequencing. And is there any evidence that certain states of people's microbiomes might provide resistance to a SARS virus or an enterovirus?
There's certainly a theoretical reason to link this. So if we go back a bit, because the data at the moment on the microbiome and COVID, for example, is at the association level and doesn't really sort out causality. Certainly there's a causal effect. Your microbiome deteriorates rapidly when faced with a big COVID infection. But I believe that if you take a view that the microbiome is crucial to your immune system, that its regulation is finely tuned in how you react appropriately to a viral infection, or you overreact and get autoimmune disease, I think it is crucial. And there's a lot of evidence building up that this is via food and the microbiome. So epidemiologically, there are associations between COVID severity and obesity, type 2 diabetes, deprivation. And we've now got some data, it's unpublished, but should be in preprint soon, showing that the quality of your diet is important. So that when you did, because 1 million people kindly filled out a food frequency questionnaire for us, as I was alluding to in the COVID symptom study app. And when we've looked at that, we're seeing quality scores in the food that even when you adjust for deprivation and BMI and comorbidities, you see an effect on severe COVID that's quite significant. So if you put all those, and they were prior nutritional data and then prospective COVID in around 1 million people. So it was quite a powerful but simplistic epidemiology study, which always can be flawed. But if you put it all together, I think it does build a picture that people with a good nutrition, high quality, eating high quality foods and diets have a better microbiome, which is more able to respond appropriately and rapidly to a viral infection and reduce its symptoms and its consequences. And I think I hope this is one of the lessons we learn, and all of us realize the importance of nutrition, not just on our weight, but actually on our immune system. And this is a really understudied area, and it would have been great, when Boris Johnson was sick, if he'd actually gone to the next step and said, well, we're going to actually change this nation's health, not just for obesity and diabetes, but all these kind of other immune problems, that this country having the worst record for ultra-processed food in Europe, has to stop somewhere, and we're going to make an effort, but hasn't happened yet.
Yeah, I've been looking into the incidence of atopic illnesses in children at age 12 months, and it can reach 20%, which is incredible to this day. And clearly part of it is diet and microbiome. That leads me to the inevitable question about long COVID. And I think probably all of us on this call know somebody that had longer term symptoms. Now, there might be a lot of psychology there, but I certainly know a couple of people who seem to have genuinely chronic fatigue syndrome. So from your studies, have you got any early signals on some of those more common, I have to call it long COVID, but let's use that phrase at least temporarily. What's your view on that?
Well, with my colleague Claire Steeves, we've got a biobank now of several thousand people with long COVID that have given us their blood for DNA and genetics and microbiome. And so we are starting to do those studies and try and work out the difference between people with long COVID, short COVID, and no COVID. And it's not immediately obvious exactly what's going on here. We've done some very small immunological pilots as well, looking at T cells and B cell differences. Again, nothing striking is coming up. Others around the world have shown differences in the microbiome. We are seeing a suggestion that nutrition is important, but again, this is very early days. And I think part of the problem is that long COVID will divide into lots of subgroups. And we published our paper that Claire Steeves led. We published in Nature Medicine, showed that there are at least two subgroups, a respiratory only type of long COVID, and then this other multi-organ type of long COVID. And there's probably within that other immune phenomena that we need to subdivide. So my worry is that we really need big numbers in order to subdivide into these groups so we can really pick them apart. Otherwise, we won't see the wood for the trees. And that's why I think it's going to be a tough nut to crack until we start doing this. And because the Zoe app had prospective symptoms, we were able to see that people who had in that first week more than 5 symptoms were the ones that went on to get this. It suggests that the immune system was reacting in a different way to the people that had a shorter version of it, and why your nervous system would react when you anosmia at the same time as your muscles and your gut. I think this really should be telling us that something that fundamentally at the immune level is happening. But we are, as you know, our tools are quite crude for looking at particularly T cell differences in large numbers of people that we need at the moment. So I think we'd be looking at some sort of fix, and it could be the microbiome might be a sort of surrogate that you can do in thousands of people to look at this. But yeah, it's early days. I mean, we've been studying autoimmune disease for 100 years, and we're still puzzled by them. We still use steroids in everybody. So it's not going to be an easy one to sort out, but definitely collecting the right data really is crucial. And I think the detailed phenotype, I think, is really what we need, and that's what we're continuing to collect through this subset of the app, which is the bioresource.
And I was asked this question recently: is the microbiome in an individual seasonal? Can you get serial longitudinal measures, and is it different in the winter versus the summer in a person?
Definitely is. They've done some studies in Africa or Asia where they had the wet season and the dry season, and showed clear differences in those populations, even on very small numbers of people. So I'm sure for us there will be differences as well, even in the UK, where it seems to be always rainy and damp. There'll be subtle differences depending on how much time you spend outside, but also clearly there'll be, if you eat seasonally as well, you'll see those differences. So there's day-to-day differences, there'll be seasonal differences, there'll be some health differences, and also there'll be that unique signature that is unique to all of us, that runs through the core of us. So I think we need to start separating those lines out as we look more and more longitudinal studies to work out what the core is. And that's why it was quite helpful to do these large metagenome studies, to say what are the 30 microbes that everyone has that you can actually relate to, rather than just picking out random rare things that are hard to quantify.
Back to diet again. So somebody's asked, you're all on all these seeds and nuts and red cabbage, but you're eating less of other foods that might be good for you. I'm going to say red meat, because that's what the questioner has asked. What's your view on that? That you go over the top and you're missing some other food that's actually good for you, and good for you because you're that person with that genetic and environmental profile.
Well, my advice is eat as much of everything, eat a variety of everything. So, I eat meat once a month, and so I'm not a pure vegetarian, and I'll pick a nice bit of grass-fed beef and have that once a month, because I believe that there might be vitamins and nutrients in that aren't in purely plants. And for the same reason, I have fish a couple of times a week, despite seaspiracy, line caught and local boat. But I think that the principle is that I think variety is key, and I'm not telling anyone to have just kale 3 times a day and then a kale smoothie as a treat. I think we've got to get away from this fad diet idea or keto diets that really limit what you're eating. The key to a healthy microbiome is to have diversity, and so it doesn't mean you don't have an egg once a week or whatever. I don't think you need to have meat every day. I don't think you need to have an egg every day to be healthy. But I do think you need a variety of plants and in small quantities to do that. And I do think you benefit by having fermented foods on most days as well. But red meat, interesting, there is a personalized nutrition area of red meat, which we're going to start studying because there is this whole research about TMAO and red meat and heart disease, that some people have the microbes that convert TMA to TMAO, which supposedly gives you atherosclerosis, whereas others don't. In the future, if we know what the microbes are that are doing that, we will be able to advise people about potentially limiting the amount of red meat they have more than others. But in the future, we're going to be having stem cell meat anyway, so we can manufacture it that it's low in choline and so won't cause any problems.
Meat on a dish. So I'm sure lots of people are analyzing the microbiome of people over aged 100 years. And have you touched on that area? There's all sorts of rumors that people that live that long, for example, in the Japanese, might have very healthy microbiomes. Have you investigated or thought about that?
Well, the microbiomes of people who are alive are always better than the ones that are dead. I think that's my general rule. And this longevity research is always a bit biased by that effect, because we nearly every disease, every comorbidity affects your microbiome. Results on the 16S, that's the early way of doing the microbiome, suggested there was like a cutoff at about, there was no real difference until about the age of 70, and then it sort of fell off a cliff. With the metagenomes, we're seeing a more subtle picture that actually the diversity or richness does slowly decline, as with other blood markers and biomarkers. So I doubt if there'll be a 100-year-old microbiome that's going to go on sale for transplants. Although it's interesting that, perhaps keeping your own microbiome when you're a fit and healthy 30-year-old, stick it in the freezer, as they do at cancer hospitals for people having immunotherapy, etc., would be a thing that would be useful. So if you are feeling healthy and you've got spare place in your freezer, it might be worth giving it a go.
So you've partly answered this during your talk, but the people living in the same household presumably do share a bit more microbiome. People who have pets, I mean, there's been a huge resurgence of people buying pups, and presumably it does change people's microbiome if they live in the country on a farm, or they live in a densely packed city, or they've got pets or they don't have pets. What's the results on that so far?
There's definitely a rural versus urban difference, and we've seen that in our twins, that there's greater diversity of microbes of twins living in the countryside rather than in urban areas, and other studies around the world have shown that as well. So being in close contact with soil and green sites is really important. Having a pet, but mainly dogs rather than cats, increases the diversity of your gut microbiome, which interestingly seems to be inversely related to allergies as well. So there's a sort of parallel between the epidemiology of the microbiome and these allergy patterns, which is also quite interesting. So when we looked at our maps, we also saw regional differences, which, difference between the north and the south of England is really quite striking. That different microbes live in different environments, and we don't think we can explain that just because of regional cuisines, Lancashire hot pot versus pasties, or etc. So it's something other in the soil, in the air. Air pollutants might also play a role, and we're looking into that in our group as well. So I think we have to keep an open mind that our microbes are perhaps more sensitive than we think. They also respond to things that are all around us, like pesticides, herbicides, like glyphosate, that is in all non-organic foods, and in different areas of the country have quite different amounts depending on whether they're spraying a lot of it. So I think it's opening up a whole new area of environmental medicine as well. And another reason we're all quite different.
Yeah, the incidence of type 1 diabetes in Britain varies by 80-fold across different regions, and it remains unexplained. It's not all genetics by any means, and there's a rural versus urban effect there that needs to be explained and then applied to better health. There is inevitably a vitamin D question. Now, you were one of the leaders in the Sunlight Consortium that led to the first GWAS analysis of circulating vitamin D concentrations. And that's, of course, what you and George used in that MR study recently to say there wasn't much of an effect in type 1, but it looks as if the multiple sclerosis results are more convincing that higher normal vitamin D might be protective. But Gavin McKinley's asked, what about vitamin D and impacting the microbiome?
Vitamin D has ended up being the thing I'm most hated for. Because, of all the things I did, I spent 20 years trying to show how good it was, and the last sort of 10 years trying to retract it all, and get a lot of flack every time, particularly in the nutrition field. People are very passionate about their sunshine vitamin, but the Mendelian randomization has confirmed all the clinical trials that actually, for its use in bone health, it's completely useless. And I think unless you're kept in the dark in an old age people's home, that is, which is really interesting because that's why I got into it, and most people take it. I think with the exception of multiple sclerosis, where there is some evidence, it's all pretty weak and likely to be confounding and mixed causality. But and of course, I went out and said, well, I didn't think it had a role in COVID either, despite sales having gone up 400%, and lots of companies, retiring on the proceeds. So immune busting effects,
right,
which are pretty hard to find in the literature in any clarity, other than in a few association studies. So the microbiome, vitamin D. I come from our question now was vitamin D in the microbiome.
Yeah.
I don't think there's a relationship.
All right.
Is all I can say on that one. Just go in the sun and have a varied diet with mushrooms, and you get all the vitamin D you need.
So we've only got 4 minutes left. Two very quick questions. We've had Jamie Oliver, we've had the footballer, and we've had Boris announcing a campaign against obesity, including childhood obesity. What in your view, what should we do about essentially this crisis that we've got in obesity and poor health? How do you make it happen? I guess government has to do this. Policy has to change. I guess Teresa Marto touched on this earlier today.
Yeah, I think we have to recognize what the number one problem is with this country and its health, and focus on that, and bring food quality into the equation, because we need to get rid of our discussions on calories. We need to get rid of our discussions on fats and carbohydrates. We just need to say, in this country, we eat more ultra-processed crap than any other country in Europe. We're second only to the US, and it's getting worse. And it's getting cheaper relative to whole foods. And studies are now showing that you have ultra-processed foods, you're going to overeat by 20%. And if you just took that and extrapolated it, it's not hard to see, even if you don't understand the mechanisms. If you just took that figure from that NIH trial, you just say, okay, just based on that, let's convert this country from being the worst in Europe to being the best. How do we do that? How do we educate everyone from children upwards about food quality and start investing in that? That is what I would do, and that's what Boris should do. Stop pretending that we can fix it just by sticking some labels on saying this is low calorie processed rubbish that's going to get us even sicker. I think that's it, and we need to copy places like Portugal, Italy, that have just said no to ultra processed food and have much better rates of health than we do.
Yeah. So Tim, I hope everyone has enjoyed this. Thanks for everyone asking questions. Thank you, Tim. I must say I've really enjoyed the opportunity to talk to you and put forward these questions. These video calls, you usually run out of time to get any questions. So that discussion aspect, we've lost a lot. We'll probably get it back later in the year or next year. We're looking forward to that. So Thank you very much indeed. And I'm sure that you'll get thousands of views on YouTube coming up. Thank you, Tim. Thank you very much.
Been a pleasure.
Good evening. Good evening, everyone. Thank you.